So you want to be a biochemist? Here are some of the basic things that can get you off the ground running. First off, memorize your amino acids and your word roots. These are going to come in super duper helpful. Thankfully, there's not a lot of stuff that you're going to have to memorize, but there's a lot of different softwares that you're going to have to learn to use. Those amino acids are going to come in really helpful when you're talking about proteins. And so when you're talking about proteins, you want to get familiar with UniProt, as well as the protein data bank, the PDB. You're also going to want to learn a molecular visualization software, something like PyMOL, ChimeraX, MolStar. I would recommend either PyMOL or ChimeraX if you really want to get into the biochemistry world. Nucleic acids, familiarize yourself with NCBI Nucleotide, which encompasses things like GenBank and RefBank, and it has BLAST, where you can basically find similar sequences and compare sequences and things. You're also going to want to familiarize yourself with some sort of molecular biology tool, something like Benchling or SnapGene or something like this, in order to manipulate plasmids and look at plasmids, design primers, all of these various things when you're thinking about those nucleic acid sequences. You're also going to want to learn some sort of vector graphics software, and so Adobe Illustrator, or a free version is InkScape. What these will do is they'll allow you to make graphics that then you can blow up really big and say on a slide projector and not lose the resolution, because you can save them as things like PDFs, or what you could do is you could just save and export really high resolution versions of things. Speaking of slideshows, you're going to want to get really used to making PowerPoints or Keynotes, presentations, this sort of thing, and have versions saved to them that then you can go back to and modify and expand upon. Setting up your computer, you're going to want to make sure you're really organized, and so you're going to want to have some sort of cloud storage typically, so whether it's Box or OneDrive or something like this, where you have folders that are organized for your different types of experiments, say, or by date, these sorts of things. You also want to start off establishing a really good naming system for your files, and so I recommend that you put the date in your file names and you don't use any spaces, instead use underscores, and this is going to allow the computers to be happier with you, and having the dates is going to allow for easy cross-referencing between things. I also recommend keeping an electronic notebook, and so go ahead and get that started and set that up. Get used to using spreadsheets and making master mixes and doing calculations in spreadsheets rather than just writing things out by hand, and this can save you a bunch of time and allow you to reuse the things from one experiment to another experiment. Speaking of reusing things, you're going to want to be able to copy and paste things that you've copied a while ago, and so I recommend installing something like CopyClip, which is a clipboard manager that is then going to allow you to kind of copy something and then find it again and paste it later on. Speaking of managers, you're going to want a reference manager, a citation manager, something like Zotero or Mendeley. This is going to hold all of those scientific papers that you're coming across. You can even annotate in there, and it'll keep the annotated versions, and it will allow you to quickly create bibliographies from those. Speaking of bibliographies and stuff, you're going to want to get really good at reading scientific papers. It takes a lot of practice, so get started with that practicing. And then be sure to save those papers into your library. To find papers, you're going to want to familiarize yourself with things like PubMed, Google Scholar, looking at where things have been cited and going to find the papers that have cited the paper that you're on right now to see what's kind of come from it after that, and to look at what the paper you're reading is citing in order to find things like review articles. You can set up Google Alerts if there's a field that you're interested in, a topic that you're interested in, an author that you're interested in. You can actually go and set alerts so that it'll tell you if that author publishes something or there's a paper related to that paper that gets published or something like this. Keep notes with your papers. Get really familiar with librarians at your library. Librarians are awesome, and make use of things like interlibrary loan. If they don't have an article or a book that you want, they can actually go and get those for you, and they can also help you with your research, and they're really typically very, 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 very nice people. So librarians are very undervalued. So those are kind of the main things that will get you off the ground running if you want to start a career or just test out a career, whatever you want to do in the biochemical sciences. And so again, you're going to want to do amino acids, word roots. You're going to want the Clipboard Manager, the Reference Manager, learn the PDB, UniProt, NCBI Nucleotide. You're going to want PyMOL or ChimeraX, some sort of molecular visualization software, some sort of molecular biology tool suite thing, SnapGene, Benchling, CLC Main workbench. There's a variety of different ones, APE, all these various tools that will allow you to look at nucleic acids and design primers and things, and get used to reading papers, finding papers, tracking papers. Make use of the mentors around you, find a supportive lab, and remember to have fun along the way.